also then going to trigger the G-alpha subunit to exchange GDP for the more active form, which is GTP, shown in red. Now, once you have GTP bound, that is signaling that we are in an active state, and that's actually going to cause the G-alpha subunit to separate and keep the beta-gamma as a dimer. Again, they're still going to be associated with the plasma membrane through the gamma portion specifically. And now we have a signaling cascade that can occur on the intracellular phase because we have this G-alpha S that's activated at this point. And that'll lead to that whole cascade of responses. Keep in mind again that there's always going to be an excess amount of GTP over GDP in the cells, and so it's always ready once ligand is bound, right? So you don't have the availability of GTP being a rate-limiting step. Really, a lot of the rate-limiting step is going to be the interaction of the ligand or drug with the receptor. So again, just as important as turning on these receptors, we need to have effective mechanisms to turn them off as well. And so what you can think about is we're going to essentially kind of go in the reverse in this case. Now the the G-alpha protein, I should say, the G-alpha protein also has enzymatic activity in it, specifically its ability to hydrolyze GTP into inorganic phosphate and GDP, okay? So once you have that hydrolysis occurring, you lose that phosphate group here. You now have the GP bound inactive form, and that is going to lead to a conformational shape change that will allow the reassociation of this heterotrimeric protein complex here. It's now to be in the inactive form. So one thing just as a reminder, we talked about inverse agonists previously. A number of GPCRs are going to be constitutively active. So they won't always need that binding of the ligand on the extracellular surface. You can have inverse agonists that can tamp down some of that endogenous activity as well. So I just want to keep that as a caveat from what we said today and in the videos. Now, what's neat about this whole process is relatively slow when we think about the whole time frame of signal transduction. And by having it be relatively slow compared to some of the on, is that it allows, when you have this G-alpha protein in the active form, it can trigger and hit a number of molecules intracellularly. And this is where we start having that amplification of the signal. It's not like it's going to interact with just one protein kinase A. It can interact with multiple of them as they go along and activate some of the intracellular signals. And suffice it to say, I feel like we often neglect the beta-gamma dimer, but it does have some intrinsic activities itself. Of course, through that lipid moiety that's found at the end of the gamma subunit that's going to help to anchor this to the plasma membrane. These 20 carbon and 15 carbon geranyl and farnesyl groups that are going to bring it there. We also are going to be able to get a lot of our heterogeneity of the different GPCRs by having different kinds of beta and gamma subunits that are going to couple to different GPCRs. That allows some cell-specific selectivity, some tissue-specific selectivity. They're not all using and competing for the exact same combinations of beta and gamma subunits. We know that some of its functions are going to include localizing, coupling, and deactivation of the alpha subunit. It's also going to function to help reduce the tendency of GDP to leave the alpha subunit, kind of that negative feedback. Can also regulate how well ligands are going to bind to the GPCRs. Can regulate some phosphorylation of receptors by specific kinases. And they also can act as signaling proteins. They can enhance potassium and decrease calcium channel activity. And this will kind of come back to after we look at the ion channels next week with Dr. Gao. So let's look at a little bit more of what happens once we have this activation that's going to be occurring. So again, we have our ligand-bound receptor up here. You're going to then we have the activation of GDP to GTP. And now we can see again that we have the G-alpha subunit here that's going to be interacting with adenylyl cyclase. This is a G-alpha S in this case because it's going to enhance the conversion of ATP into cyclic AMP. Again, the beta-gamma dimer is over here waiting for the hydrolysis of GTP to occur. Now, once you have the cyclic AMP that's being made, this is going to be able to convert the inactive protein kinase A, or pKa, into the active form. So in the inactive form, you can see we have 2 regulatory subunits and 2 catalytic subunits. The catalytic subunits are shown in green, and the regulatory are shown in blue. You have the binding of cyclic AMP specifically on the regulatory subunits. And the binding, in this case, is what we call cooperative, because you're going to actually need to have 4 molecules binding to these 2 forms of the regulatory subunit. But once you get that initial binding with cyclic AMP, there are conformational changes that are going to occur in these regulatory subunits that actually make it easier for the other 3 to be able to bind. So kind of just getting that initial activation energy to get that first one bound, then will make it easier for the other 3 to bind. Once you have these occupied by cyclic AMP, that's going to allow for the liberation of these catalytic subunits that we can see in green, and they're going to be in the form of monomers in this case. So now you can see these 2 activated protein kinase A catalytic subunits. They're going to be able to, what we call, translocate. They're going to go across the nuclear membrane by going through nuclear pores within the membrane of the nucleus. And these activated protein kinase A, they're going to phosphorylate. That's their job. They're kinases. That's what they do. They add phosphate groups to target proteins. And one of the important ones is going to be CREB, which is the cyclic AMP responsive element binding transcription factor. We just call it CREB. Once you have CREB going from its inactive unphosphorylated form to its phosphorylated form, that's going to allow for the recruitment of a number of different proteins involved in the transcription of genes. So most importantly, RNA polymerase is going to get recruited, but then as we'll talk in 2 weeks about coactivators and other accessory proteins that we need in order to allow transcription to occur. Once we have transcription, that means we have made new mRNA molecules, and then those mRNA molecules are going to be converted into proteins. And so that might be different enzymes, cytoskeletal proteins, other receptors, lots of things that can happen as a result. Let's do a few poll everywhere questions. These are not from your homework. These are just kind of from what we just talked about and what you saw in the videos. So all of the following are primary messengers except which one? A couple more seconds. Okay, excellent. So we've got calcium. Again, that's going to be more of a secondary messenger. We'll be talking more about, sorry, I don't know what I touched. Secondary messengers when we get to the intracellular signaling, and then kind of throughout all of these lectures on GPCRs and ion channels as well. Great job. Let's do the next one. So in the video, we talked about 3 different types of G proteins. And right now, we just talked about one of them. Which one is going to decrease cyclic AMP levels? Couple more seconds. Great job. So J alpha I, right? So essentially it's kind of the opposite of the signaling scheme that we just talked about before. And as we go through pharmacology and the modules next year, there's going to be different receptors. So especially when you start off in the fall, and Dr. Gao says, okay, you know this receptor works through G-alpha S or works through G-alpha I, then he will know that you already know all of the downstream signals as a result of that. And as we talk about different classes of drugs, some are going to be working selectively on G-alpha S coupled, and some will be working selectively on G-alpha I, and some will be non-selective, right? And then it's kind of them battling it out. And then we'll also talk about G-alpha Q on Wednesday. I think there was a question in the middle.
You just answered it.
Okay, good, excellent. Good. Getting ahead of it. Okay, next question. Which of the following functions of the beta gamma dimers is incorrect? What do you guys think? Which one is incorrect? Excellent, great. So that's going to be the alpha subunit, right? That we talked about will be the GTPase activity. Excellent. I think we've got one more question before we get into the homework. So which of the following forms of protein kinase A is correct? Which one is correct? Okay, before I show it, I'm curious if anybody on this side of the room would help us answer this question. What does anybody think it's trying to answer? Maybe not on this side of the room, but we'll put your hand up first. Go ahead. That is correct. You get one too. Just need more caffeine. I gotta hand it up early. Great, that's correct. It's going to be those regulatory dimers bound to cycloGAMP, where we have that cooperative binding and then release of the catalytic subunits. Great job, guys. Okay, so let's pull out your homework. Again, the pull everywhere is going to be pretty fast for these because the assumption is you've already done these questions. If you didn't do them, pick something. Okay, so I'm going to open up the first poll, so you can go ahead and get your answers in while I read the case. L-deglutide is a long-acting form of the glucagon-like peptide GLP-1, which is used to treat type 2 diabetes. Just take it down for a little bit, just want to read the case. Thank you. It binds to and activates the GLP-1 receptor, which is expressed in beta cells of the pancreas, as well as neural cells and other tissues. The receptor signals through G-alpha S. The drug causes release of insulin from beta cells, as well as reduced appetite due to signaling in neuronal cells. So which of the following would you expect to occur? Just a couple more seconds, get your answer in. Okay, let's see what you guys Okay, excellent job, guys. So we have the correct, sorry, I'm touching something. We've got the correct answer there. So binding of GTP to the alpha subunit is going to be needed, right? Because reducing appetite is the pharmacological outcome that we're looking for. And we know that we need to have GTP bound, in this case for G-alpha S to work. Let's go ahead and go through some of the other ones. Beta cells will have dimers of pKa bound So remember, that was our last question we talked about before. The regulatory subunits bind to cyclic AMP, not the catalytic. Let's go to the next one. So again, because CREB is a transcription factor that's getting turned on by G-alpha S, that means it's going to increase transcription. And then let's look at this last one. Beta subunits will activate pKa and production of cyclic AMP. So now remember, we were going to talk about the alpha subunit is going to be important, right? That's the actual one that's going to lead to a lot of this signaling that's going to activate protein kinase A. Great job, guys. I'm going to go ahead and open the next poll so you can get your answers in while I read the question. Beta-sol is used as a stimulant to cause production of gastrin from the stomach and can be used to diagnose Zollinger-Ellison syndrome. Beta-sol is an agonist that binds to and activates the histamine H2 receptor. This interaction occurs at what part of the receptor? Just a couple more seconds to get your answers in. Okay, let's see what you guys selected. Excellent, fantastic job, guys. The hydrophobic core. So if anybody had any difficulty with this homework question, this is where it comes from. Again, we saw that there's multiple places, and it kind of makes sense. Again, we have a lot of real estate to work with here as far as amino acids that are accessible and available to ligands to bind. It makes sense that some stuff that are smaller are able to kind of go a little bit further in, and they can bind to the hydrophobic core, kind of traversing in between a lot of those amino acid residues, and that's where histamine falls. We can also have short peptides that are going to bind to the core, as well as some of these little loops that are formed as we kind of go back and forth. We also can have some that interact with the N-terminus and some of those external loops as well, kind of throughout here. Some can actually kind of rearrange the conformation of the N-terminus. And then in a very unique case of these proteinase activated receptors, or PARs, they can cleave the receptor and lead to a new form that's exposed here that acts as an autoligand. It kind of turns itself on, kind of a self-fulfilling receptor. Okay, and we've got one more question. I'll go ahead and open that poll. The histamine H2 receptor has constitutive activity in the absence of ligand through G-alpha S signaling, while beta-sol acts as an agonist of the H2 receptor. Famotidine or pepsid acts as a competitive antagonist. Which of the following statements is correct? Okay, a couple more seconds. We'll get over to sharing. Yes. See what everybody thinks. Okay, good job, guys. Very nice. So in this case, famotidine is going to reduce the dissociation of the alpha subunit of the receptor from the beta gamma dimer. Great job, everybody. Okay, so what we're going to do now, let's see what time it is. Okay, so we'll take about 15 minutes. There are cases uploaded to Canvas that I added earlier this morning. Go ahead and download those, and you're going to work on them with those questions. At least one of the questions you guys are going to have to look up the answers. It's not in your notes, so you're going to kind of use your drug information skills as pharmacists to be able to look those things up. But feel free to work by yourself, in pairs, in groups. I will be walking around and helping you check your answers to make sure you kind of bank as many pull everywhere points. In about 15 minutes, we'll come back together again. We'll kind of go through the correct answers. You'll make some more points. And again, same thing as I walk around, flag me. If you have a question about the video, about any of the questions from today, flag me. This is a great time to get your questions asked and answered. So work on that. If you have any difficulties accessing the cases, just let me know, and I can double check if I uploaded them correctly.
Where is it at?
Should be on the Canvas site for today. Is anybody else able to find it? Okay, so at least one person has it. So yeah, it should be right under the first GPCR 1 or whatever I call it today. It should be right below or above your homework. I added it this morning. Okay, it says cases, I think, and they're hyperlinked. Okay, I'll be coming around with any questions to help you check your answers. And I'll open the first poll for them too, so in case you want to get your answer from the first one.
She gives you buffer points, so it's gonna, you're probably gonna get a.
Last week and I got 100, but then I was angry and stuff.
Really?
And I got a 14 out of 16 in that class. I didn't get what's going on.
Wait, that's weird. That's weird.
Yeah, nothing is making sense.
هم مش عارفين هما بيعملوا ايه بصراحة.
Yeah, I agree.
I mean, to be fair, أنا مش عارف أنا بعمل ايه برضه بس يعني.
Hello. So, I was looking at the book for Wednesday's class. And there were two questions that were kind of bugging. It was true false. PKC is active without calcium. It was true. But isn't it calcium and DAG together?
So it can be more than one thing. So yeah, I wouldn't get too caught up in that. I think it's just more making sure you know which regulators are involved. So you know what I mean? Like, it's kind of hard to have like some nuance in the code and stuff. As long as it's one of the regulators, essentially, it's hard.
Okay. And then this I couldn't find them at all.
Gotta start working on your cases for today. There.
What did we talk about for
I should have said it. I didn't.
So, there should be something to set. Do you want to send you a picture of the case?
Teacher, you as a file, send it to me as a file.
Why does that sound to you? How do I send it to you? Dry? Dry?
Okay, sure. Send it to me. Thanks, bro. Do you know this thing called
Guys, would you say the first question? I didn't do
Intracellular.
Yeah, I know. I found this. Testosterone. The other one.
Testosterone is intracellular, so it's inside.
I'm just gonna take a guess and say,
Let me answer.
no.
Then the second one is basically activity. Maybe cell surgery. Yeah, I'm gonna say it. Guys, by the way, how you guys studying for pharmacology?
I really don't know.
My biotech is exactly 99 flashcards. And most of them are, look at this pic.
Oh, but is that the whole?
Oh, it takes. You're saying A from
I don't know. I think. I don't know.
I'm gonna answer it.
For me. For me. Okay, amazing. But for me,
We haven't even
What is that?
I don't know what you're about.
It's an electric advance from undergraduate.
With the rest of the team. So many names.
My mom doesn't like it. On me. Even at work. Even at work. This is like, somebody was scared. I was like, this is like, you don't like it. I'm trying to switch around. I was like, I want to do different. I don't want to work. I couldn't listen to that when I asked you. But it was, I couldn't eat flashcards. Like, I studied.
No, do it. For every view, we get $5. For every view, you get $5. A paid subscription, $7.99. Wait, I don't want to pay for it, to be honest.
Okay, let's go ahead and put the answers in for the first question. The poll open. So the first question was primary messengers interact with receptors in various cellular locations. For example, the hormone testosterone interacts with its receptor in one spot, and the vasoactive chemical histamine reacts with its receptor either inside or outside the cell. Get a couple more answers in. One student. Oh well. Oh, you're good. Okay. Lockett, who would like to answer this question? So which one? The first one? A, yep. So the first one is correct. Great job. So in this case, remember, most hormones are going to work on those nuclear receptors, right? The nuclear hormone receptors. And because they're quite lipophilic, they can cut across the lipid bilayer very easily. And then they make their way to be able to access either cytosolic or nuclear proteins, but that ultimately are going to end up in the nucleus to allow gene transcription. And then again, we've spent a fair amount of time today already talking about histamine. And we like histamine is going to work on GPCRs. And as we said in the beginning of class, the ligands for GPCRs are always going to be on the outside of the cell, extracellular. Or like I said, a little bit can kind of dip into the lipid bilayer, but none of that ligand binding is going to be on the inside of the cell. Great job. Okay, let's go ahead and open the second poll while I read the question. So the next receptor we talked about here was the vasopressin receptor, or ADPR2. We talked about vasopressin last semester in patho. You know it's found in the distal tubules and the collecting ducts of the kidneys. Actually, we talked about physiology too. We know that when you bind the ligand, which is either called vasopressin or antidiuretic hormone, or ADH, it's going to cause signal transduction that's going to lead to the insertion of proteins known as aquaporins into the apical membrane and reabsorption of water. We know that this receptor is a G-alpha S, and so which of the following would be the correct order for signal transduction? Get your answers in. Okay, go ahead and block it, Tony. Who wants to answer this one? It's like a tie between the two of you. I don't even know who to pick. I feel like Harry's favorites. Okay, who wants to answer this one? I already forgot the question. Yeah, that's correct. You guys both get that. Great job. So D is the correct answer. So you're going to have after the ligand has bound, you're going to have binding of GTP to the G alpha subunit, in this case is S. That's going to then lead to activation of adenylyl cyclase. Adenylyl cyclase is the enzyme that makes cyclic AMP, so levels will go up. We're then going to have a release of the protein kinase A monomers because the cyclic AMP has bound to the regulatory subunits, that cooperative binding we talked about. We now have the catalytic pKa can go into the nucleus, and that's going to enhance transcription, or in this case, can increase aquaporin 2 insertion, which is the final physiological or pharmacological effect here. Great job. Okay, well I just want to give you like flashbacks.
I remember
from
this.
half oh my God, information is coming back.
Then affected phase. This
This
sounded
is what
notes.
we were coming back to, where the vasopressin receptor and the insertion of those aquaporins into the membrane. I'm sorry, that was very neat to do, you guys. But you're going to see again next year too when we get to some of that pharmacology and cardiology, renal. Okay, question 3. There's an outbreak of whooping cough that was reported in Alberta, Canada due to vaccination rates dipping below the 98% required for herd immunity. We know that whooping cough or pertussis is caused by an infection with the Bordetella pertussis bacteria. The pathogenesis of this infection is due to pertussis toxin. So what is the effect? How does this interfere with GPCR signaling? Let's see what we learned. Yes.
So it blocks the G alpha I subunit.
Yes, so the bacteria is going to block the G alpha I subunit. So think about what would that mean if you were blocking the G alpha I subunit. So G alpha I, as you guys know, is the negative pathway, right? That usually reduces cyclic AMP. Sorry, I don't know why I'm getting feedback. And so instead, because they're like a negative of a negative, we're now going to actually have higher levels of cyclic AMP. Okay? Higher levels of cyclic AMP. And so that's how this is going to interact. So a fair amount of these toxins that are released by bacteria and other species can actually have these responses. Great job. So it's blocking G alpha I. Let's do one more question. So, I guess I was supposed to do it as a poll. I forgot I put it as a poll. Don't worry about it. And so we'll activate the last one. Okay, I just wanted to give out prizes. Okay, so the last question, now we're going to be talking about a different toxin here. So we know that the CFTR, this is the cystic fibrosis transmembrane receptor. This is the receptor that's mutated in patients that have cystic fibrosis, but it's also, as you can imagine, very important in normal homeostasis. It's a chloride channel that secretes ions, not only chloride, but other electrolytes, into the lumen of the gastrointestinal tract, right? So that's important to be able to have salts as part of our salt excretion. If you have an infection with cholera, the bacteria Vibrio cholera, there's going to be massive loss of salts from intestinal cells, which causes water to diffuse into the lumen, causing life-threatening diarrhea. So what do you think is the likely mechanism by which CFTR is targeting cholera pathogenesis? So you can kind of think about what's happening here. This is where we are putting our answers in.
I mean, based on the question, it's letting in too much ions. So
I'll take your word for it.
something activated, so it can't be
I need to think
the
about the
values
question.
A, C, D, and E are all activated.
There's a couple more of you who want to make sure.
Are both POC pathways.
And then we'll review how this is going to happen.
What's
Okay, you lost 3 students.
Whatever.
It's okay. So you're fine with that.
I'm curious.
Okay, let's see what we already picked. So the vast majority of the class has the correct answer, but let's take a look at this in a little bit more detail. I will add this slide to Canvas so you guys have this as well. Let's look at how does this work. So again, a cholera is going to have a toxin that's going to be released. In this case, it's going to bind the G alpha S subunit. So you can kind of see right here, we have the cholera toxin right here. It's going to bind to its receptor here. And what ends up happening is by binding to G-alpha S subunit, it's going to then be able to increase the activity, and it prevents the GTPase activity. And then that's going to lead to higher levels of cyclic AMP being made by the adenylate cyclase. I'm sorry, we have the cholera-toxin here. Here's the G protein down here. Adenylate cyclase, make lots of cyclic AMP, and cyclic AMP is going to then turn on PKA, and PKA is a regulator of some of these channels that we just talked about, like the chloride channel here. And so in this case, you can see the CFTR right here in particular. And if you think about having a lot of electrolytes going into the lumen of the intestine, we know that, just like we talked about in patho last year, water is going to follow where the electrolytes go to kind of dilute out all of those electrolytes. So if we have activation, persistent activation of this pathway, a lot of chloride and bicarb and things going out there, the water is going to passively follow. And this literally, I know we've all probably had diarrhea at some point. That is not the case when it comes to cholera. We are talking about life-threatening diarrhea because you're going to be so dehydrated, you're losing so much salt, it's going to lead to arrhythmias and then lead to death as a result. So it's a really serious type of infection. So I'll pose this last question. Yes, this last slide.
I saw that it said on the PKA slide that it has a lot to do with cellular transport. Is that just for ions, or does it transport other things as well?
So there's going to be a lot of targets. So in this class, when we think about these protein kinases, we're going to circle back to this in the very last lecture before the exam, because we're going to start talking about the interconnectedness of a lot of these different pathways. And so we're not going to cover all of the things that these molecules can regulate, but certainly protein kinase A is going to regulate a lot of channels, gene transcription through changing transcription factors. We're going to be talking about other subsequent kinase pathways it can regulate as well. And so especially after Dr. Gao teaches ion channels next week, then we'll start to look at kind of that cross-regulation. So even though we talked briefly about kinases today, we're going to cover them in a greater depth. And so you're going to see as you sit down and look at all of the signal transduction lectures together, some of it that we're kind of talking about this week will make even more sense as we kind of go into a deeper dive into some of these different pathways and how related they are. Yep, it's going to definitely be able to regulate ion channels. Okay, great job, everybody. Please come get your prizes. There is no tren or tuition or
somebody said tren?
But I put some highlighters in there, and there are Legos.
It's a steroid.
There's I put more gum in there. So have a great day.
As far as I know, tren is steroid.
Interesting. You wanna come? I don't like anything. I'm gonna go to you.